Eve was created from Adam's rib. None of these explanations can possibly be actual description. Human beings are mainly water, not dust, and there is no process by which an adult person can be made quickly from a rib. What? Seriously? Like, the fact that I even have to read that is just sad. I want to say, literalist readings of Genesis imply that God specially created Adam and Eve, and that all humans are descended from these original parents. Such readings, unfortunately, do not fit the evidence for several reasons. Okay, so they would say, well, this is how the Bible reads, right, that Adam and Eve were the beginning, right, of all mankind. But, right, there's always that, but we can't believe that because of what the science says. The chapters of Genesis are not a literal or scientific description of historical events, but a theological statement in an ancient idiom. So why not take Genesis as it's written, right? Because of what the evidence says, because of what the science says. The last quote I'm going to share with you here comes from a book called The Atom and the Genome that was co-authored by Dennis Venema. He's actually a professor of biology here at Trinity Western University up here in Canada, and Scott McKnight, who's a professor of New Testament at seminary in Illinois, and they said this: we can be confident that finding evidence that we were created independently of other animals, or that we descend from only two people, just isn't going to happen. So in other words, we can be confident that what the Bible says isn't true. That's essentially what they're saying. Some ideas in science are so well supported that it is highly unlikely new evidence will substantially modify them, and these are among them. We can know both of those things equally. There's so much science that supports it, the observable as well as the unobservable. Okay, he say there's the science shows this, the science supports this. Nothing else we find is going to change this fact, right? They're saying both of these things are fact. They're equating them. And that Christians, they want to encourage Christians to embrace that. So, anytime we write DNA, we write it with 4 letters, A, C, T, and G, that represents the 4 bases of DNA. And so we're going to say human DNA, that series of letters on the top is human DNA, and on the bottom is chimp DNA. Now, we have over 3 billion base pairs, and so when you put it into a computer and you ask the computer to align it, there will be areas that align, okay, that are quite similar. They're not identical, but they're quite similar. And so you can see that here. Now, outside of those aligned regions, okay, there's literally hundreds of thousands of bases outside of those aligned regions that don't line up at all, okay? Those are called the unaligned regions, and they don't count those when they do the final count for the differences. There's another type of difference they don't count either. These are called gaps, or indels is the fancy scientific name. So it's areas where you have human DNA but no matching chimp DNA, or you have chimp DNA but no matching human DNA. Okay, so they don't count those either. Now, the next legitimate question in your mind is, well, why not? You know, I mean, we can see this is an observable fact, right? This is something that the evolutionists would agree with me on. They wouldn't have any issues with it. This is something we can see in the present. So why not count all the differences? Because there's too many, okay? There's too many to fit in there. I'm not lying. There's too many to fit in the evolutionary time frame. It doesn't work. No evolutionary geneticist in their right mind is going to believe that many differences occurred in just 6 million years or 7 million years since we evolved from that common ancestor with the apes. It's just too much. So you have to deal with that data, right? So let me tell you 2 ways right now, at least there's probably more, but these are the 2 common ways I see. How do they deal with it? Well, one is to make the common ancestor older. Okay, so they push the common ancestor back to 13 million years. Okay, so instead of just 6 million, it's 13 million. So that gives you more time, right? And time allows you more time to have more differences. So they can try to account for that and make that work. The problem is then that the DNA evidence isn't lining up with the radiometric dating data, with the fossil data. So all the lines of evidences are not supporting the same story about the past, which is probably an indication that your story is wrong, okay? If it's true, why aren't all the pieces of evidence lining up, right, to support it? So it's very problematic. So another way that they've dealt with this, and I'm going to talk more about this later after lunch today, is that the differences between human and chimp DNA are in parts of the DNA that are not important, okay? So in case you're not aware, in humans, only 2% of your DNA codes for proteins. The other 98% is affectionately referred to as junk, okay? That's the official scientific name, is junk DNA. And they say, well, the differences That's junk DNA. It's from our evolutionary past. It's a fossil in our genes. It's not important. So those differences are there, so it doesn't matter. Well, as I'm going to show you later, there's no such thing as junk DNA, okay? We know that, surprise, right? It's not a surprise to us as creationists, because we would expect that, right? We would not expect God to create us with 98% of our DNA that's not doing anything and just sitting there, right? Count something, and they really count just one type of difference. It's the only difference they think that makes all the difference, right, between humans and chimps. And those are called substitutions in aligned regions. So these are areas where you might have, you see them in the yellow here. So you might have like a T in the human, but you have a G in the chimp, or you have an A in the human, but you have a G in the chimp. Okay, so you have DNA in both. They're in the aligned regions, but they're different. Now, do they know experimentally that it's these differences that make all the differences between humans and chimps? No, they have no idea, right? But the amount fits their timeline, right? Because it's reasonable to have these differences, this 1 to 2% difference in that timeline of 6 or 7 million years. Now, I want to share with you this short video, and it was produced with the help of experts in evolution, okay, because I looked up the background on it. And I think they really summed up the differences between human and chimp genomes very, very well. And I will just say I appreciated their honesty.
It's often said that we humans share 50% of our DNA with bananas, 80% with dogs, and 99% with chimpanzees. Taken literally, those numbers make it sound like we could pluck one cell from a chimp and one from a human, pull out the tangled bundles of DNA known as chromosomes, unroll each one like a scroll, and read off two nearly identical strings of letters. But in reality, the human and chimp scrolls don't sync up so easily. In the 6 to 8 million years since we split from our last common ancestor, chance mutations and natural selection have changed each of our genomes in radical and unique ways. Two human scrolls fused, leaving us with 23 pairs of chromosomes to chimps' 24. Other large mutations revised huge sections of text, duplicating a chunk of human DNA here, erasing a chunk of chimp DNA there, while throughout the scrolls, tiny mutations swapped one letter for another. When researchers sat down to compare the chimp and human genomes, those single-letter differences were easy to tally, but the big mismatch sections weren't. For example, if a genetic paragraph, thousands of letters long, appears twice in a human scroll, but only once in its chimp counterpart, should that second human copy count as thousands of changes, or just one? And what about identical paragraphs that appear in both genomes, but in different places, or in reverse order, or broken up into pieces? Rather than monkey around with these difficult questions, the researchers simply excluded all the large mismatch sections, a whopping 1.3 billion letters in all, and performed a letter-by-letter comparison on the remaining 2.4 billion, which turned out to be 98.77% identical. So, yes, we share 99% of our DNA with chimps if we ignore 18% of their genome and 25% of ours.
Said, well, there must have been a fusion of the chimp chromosome that occurred in the line that led to humans, which is why we have 23 and chimps have 24, right? And in fact, you know what? This idea is so embedded in the scientific minds that they actually renamed chimp chromosomes 12 and 13 as 2A and 2B, because they believe they're representative of a single chromosome in humans. That's how far this has went, right? It's actually changed the naming of the chromosome. So they believe a fusion occurred. And since all humans come from the human ancestor, that line that broke off, all humans have 23 pairs of chromosomes, okay? They always have, and they continue to have that. Now, in the chimps, that fusion did not occur, all right? So they have 24 pairs just like the common ancestor. And so since all chimps come from the chimp ancestor, all chimps today have 24 pairs of chromosomes. Does that make sense, right? Common ancestor has 24, break off, fusion occurs in the human line, doesn't occur in the chimp line. Humans have 23, chimps have 24, right? So that's why they have it today. So that's the evolutionary story that they've come up with to try to explain the differences in the number of chromosomes. And in fact, they really like this piece of evidence. In fact, they went so far as to say it's definitive proof that humans and chimps shared a common ancestor. Now, here's the thing. I don't recall, as a scientist, ever using the term definitive proof. That's a tricky thing, because we typically in science, what we're trying to do all the time is show something is false, okay? And if we can show it's not false enough, it might become a theory, and eventually it might become a law, okay? So for example, the law of biogenesis, life only comes from life. There's a few of those in biology, but not very many, okay? And so it's very, very challenging. If you're going to say something is definitive proof, it better really, really be good, right? And so I'm going to share with you a video from Dr. Ken Miller. He is a Roman Catholic, so he does believe obviously in the existence of God. And he is a cell and molecular biologist, but he's a theistic evolutionist, okay? So he believes in evolution millions of years and common ancestry with the apes. And he really likes this piece of evidence, so I'm going to let him explain it to you.
The cells of all great apes, like chimpanzees, gorillas, and orangutans, contain 24 pairs of chromosomes. If humans share a common ancestor with apes, you'd expect us to have the same number. But surprisingly, human cells contain only 23 pairs.
Now, if we share a common ancestor, what happened? Is it possible that in the line of evolution that led to us, a pair of chromosomes from a primate ancestor just got lost and just got discarded? Well, the answer to that is no. It's not possible. And the reason is because every primate chromosome has so many important genes on it that the loss of both members of a pair would be fatal. It wouldn't even get through embryonic development. So there's only one possible explanation. And that explanation is that in the line that led to us, two chromosomes that were separate in other primates became fused to form a single chromosome. And this is why evolution is a science. That possibility is testable. We ought to be able to look at our genomes and find that we carry a chromosome with the marks of that fusion on it. Now, how would we find that? It's easier than you might think.
Of every chromosome, you should find special genetic markers, or sequences of DNA, called telomeres. And in their middles, you should find different genetic markers called centromeres. But if a mutation occurred in the past, causing two pairs of chromosomes to fuse, we should find evidence in those genetic markers. Telomeres not only at the ends of the new chromosome, but also at their middles, and not one, but two centromeres. Finding a structure like this in our chromosomes would explain why humans have one pair fewer than the great apes.
If we don't find it, then the case for common ancestry for our species might be refuted. But if we do find it, it would be powerful evidence in favor of evolution. So all we have to do is to scan the human genome and see, do we have a chromosome that has these marks, telomere DNA in the center and two centromeres? Well, the answer turns out to be we do. And it's human chromosome number 2. And the evidence is unmistakable. We've suspected this for a very long time, and in 2005, the definitive study was published showing that chromosome 2 has the exact fusion point, almost the point at which the Scotch tape holds those two chromosomes together can be found. The closer we look at our own DNA, the more detailed a glimpse we get of our own genome, the more powerful the evidence for our own evolution from common ancestry with other species becomes.
Now, if you just watched this video and didn't look into it anymore, it'd be pretty convincing, right? He does a pretty good job without going into a lot of detail of explaining this. And you heard him say, there's only one possible explanation. Only one. And you know what that shows you? This is a worldview issue, right? Because I've got an explanation, right? That God created chimps with 24 chromosomes and humans with 23, right? And that's why we have that today. But see, in his worldview, there is no other explanation. There can't be. Because even though he believes in the existence of God, he does not believe in the biblical account of creation. You heard him say later, the evidence is unmistakable. right? I mean, he really, like I say, he really likes this particular piece of evidence. So again, it's really about his worldview and how he's interpreting the evidence in light of that worldview. He literally cannot imagine any other possibility. In a New York Times article, he said this: is there any question to explain these facts, right? And he says that human chromosome 2 matches chimp chromosomes 12 and 13 in light of the view that our species was uniquely designed or intelligently created. The answer is no. You can only explain this by evolutionary common ancestry. So he even goes so far as to say creation, again, that's out of his worldview. Creation is not a possibility. He says there's no way God would have done it this way. I'm going to talk to you about that a little bit more later. It has to be a common ancestry. Dr. Francis Collins, again, head of the NIH, who's also a theistic evolutionist, said the fusion that occurred as we evolved from the apes has left its DNA imprint here. It is very difficult to understand this observation without postulating a common ancestor. So he's not quite as definitive in his language, but still pretty much there. In other words, he just can't figure out how you could explain why these chromosomes match if we didn't have a common ancestor. So just to kind of review what they believe, okay, well, the evolutionary story is that in the common ancestor, there were 2 chromosomes, 12 and 13, okay, as we know them today in the chimps. But in the human line, okay, somehow very early on, a fusion occurred. And today, that chromosome is human chromosome 2 in humans. And they say the proof of that is that there's these telomeres in the middle, and there's an extra centromere. All right, so we're going to talk about that. And the first thing we're going to do is we're going to talk about the telomeres, right? So what is a telomere? A telomere is a repetitive sequences of DNA on the ends of your chromosomes. It's kind of like end caps, okay? And it literally says the same thing over and over again. GGGATT, GGGATT, and just over and over again. And telomeres kind of act like sort of a safety mechanism. So you know how at the end of your shoelace, if you have shoelaces, they have that little plastic thing on the end that's called an aglet, and it prevents the shoelace from unraveling. It's kind of a little bit what telomeres do. So because of the way DNA replication works, which I'm not going to get into, chromosomes shorten every time, okay? The replication doesn't go all the way to the end, so they get shorter. Once these telomeres get short enough, the cell will die, okay? And that's probably some of what causes aging, actually, is the shortening of these telomeres. And so they perform a very important function. They keep all the stuff in between safe, all right? That's their point. So let me point out some problems with the evolutionary story. So if 2 chromosomes fuse end to end, all right, the telomere to telomere, there should be hundreds of thousands of these repetitive sequences in the middle of that chromosome. Hundreds of thousands. They only find several hundred, right? And there's no way, again, no geneticist is going to believe you can chuck that much DNA in a few million years, okay? It's just too much. And even the evolutionist that studied this says, if these truly are telomeres, all right, why are the repetitive sequences basically at the fusion site so degenerate? In other words, why don't they look like telomeres if it's telomeres? Because they're not, okay? Because they're not telomeres. That's why they don't look like telomeres. You don't find all these repetitive sequences that you would expect to find if these telomeres had fused. Dr. Tompkins has done some research into what is there, what they're looking at and calling that, and he said, actually, it's something called interstitial telomeric repeat. So there's a really big phrase for the day, okay, scientific term. He says they're not unique to human chromosome 2. They're found throughout all the chromosomes, right? It's not special to this chromosome. They're everywhere, and they're functional, right? They have a role. They actually regulate the expression of DNA. So it's something else, right? It's not what they think it is. It looks a little bit like telomeres, but it really isn't. It does something else. Now, here's the other thing. There's a gene there, okay, at the point where they supposedly fuse together, where all these telomeres are. We know that gene is expressed in approximately 255 tissue and cell types, so it is an active gene. Here's the thing. Genes are never inside telomeres, ever, right? There's no way this can be 2 telomeres that fuse together. There's never genes there, so it can't be. So all of this evidence and more, because I don't have time to explain everything, shows that these cannot possibly be telomere sequences. And here's the other kicker to me. One of the roles of telomeres is to keep chromosomes from sticking together. So, just saying, seems a little odd. That they would be involved in an end-to-end fusion, okay? Because that's actually one of their jobs, is to keep that from happening. So off. Just seems a little off. All right, what about this second centromere, okay? So what is a centromere? Centromeres are usually found near the middle of chromosomes. They are repetitive sequences, again, hundreds of thousands of them. And there's certain proteins that associate those. And after DNA has replicated, half of it has to go, or, we have to have half it go into one cell, half into the other cell, okay? And so, and get the full complement. So they have to split it apart after it's made a copy of itself. So these little fibers come out, they attach to the DNA, and they pull it apart. And so the centromere is very important in that particular thing. So the deal with the centromeres is kind of like the telomeres. I always say it's same song, second verse, okay? Because same idea here. You expect thousands and thousands of specific repetitive sequences. We know what centromeres should look like, and you only find several hundred here, okay? It's literally 1/10 the size of a regular centromere. Nobody's going to believe you could chuck that much DNA in just a few million years. There's just too much of it missing, basically. And, once again, the sequences are not specific to centromeres, okay? They're found throughout human chromosomes, okay? It's everywhere, again. And guess what's there? A gene, right? Genes are never in centromeres, okay? And because it's just repetitive sequences. We know that gene is expressed in brain and reproductive organs, and again, like I said, they're never located inside centromeres. So if you understand the story for the telomeres, you basically understand the same thing for the centromeres. Same idea for both of those. So it is not a second centromere. So instead, these alleged telomere and centromere sequences are functional elements designed by God. They have a role. We know there's genes there. We know there's regulation happening there, and they're important. And like I say, that's just some of the problems with the fusion idea. There are more. Now, I want to share with you just one last thought from Ken Miller about this issue. And he stated that people that believe in an intelligent designer, how they would respond, okay, how people like us would respond to the overwhelming evidence, which isn't so overwhelming as we've seen, for a fusion event, and the problems that creates for people like us who believe in a creator God.
How would intelligent design explain this? Only one way, by shrugging and saying, that's the way the designer made it. No reason, no rhyme. Presumably there's a designer who designed human chromosome number 2 to make it look as if it was formed by the fusion from a private ancestor. I'm a Roman Catholic, I'm a theist. In the broadest sense, I would say I believe in a designer, but you know what? I don't believe in a deceptive one. I don't believe in one who would do this to try to fool us. And therefore, I think this is authentic, and it tells us something about our ancestry.
Okay. The only way that similarities between chimp and human chromosomes would ever deceive us and look as if it's a fusion in a primate ancestor is if we relied on man's ideas about the past, right? And we weren't relying on God's word that tells us the truth about the past. We know from God's word that humans and chimps are separate kinds. They did not share a common ancestor. We also know from God's word about God's character, and we know that he is not a deceptive God, right? He would never try to fool us. So as biblical creationists, if these similarities truly existed between human chromosome 2 and chimp chromosomes 12 and 13, which they don't, right? But let's just say if they did, right? Let me just take that argument. If they did, we would believe, yes, that they were created by a common designer God, and they have a good reason and a good purpose, right? I mean, they're there for a reason. Humans and chimps bodies are mammalian. They do similar things, so it's not maybe all that surprising or shocking. And so, again, remember, when it comes to science, what has happened in the past, our worldview is very important in how we interpret the evidence that we have today in relationship to that past. Miller is living proof of that, okay, because he has been presented with this information by Dr. Tompkins. They do talk about these things, and to my knowledge, Dr. Miller has not changed his mind. Why? Because it's not about the evidence, right? It's about a worldview. So, again, the alleged telomeres and the second centromere, they just simply don't exist in human chromosome 2. There's low numbers of repetitive sequences, and genes are found in those locations. That's just some of the evidence that shows they cannot be the result of a fusion. Therefore, it does not support, it cannot be evidence